All right, so, okay. So today we're going to really combine lectures 4 and 5, which is there's three-dimensional structure of proteins and protein structure and function. And this, it's kind of like, right, like love and marriage. You can't have one without the other. So the structure determines the function, and so we're going to talk both of these things at once. So in the last class period, we've been talking about the primary structure. And remember, it's going to be the amino acids that are in the sequence of the polypeptide that are bonded together through covalent bonds. So it's those amide or peptide bonds. And so it turns out, the primary structure is extremely important. And so if you have even one mutation of the primary structure, that is one substitution of an amino acid for something else, it could actually lead to a disease. And so one of these is, I'm going to just hold on for a second. All right. So one of these diseases is sickle cell anemia. And this is a genetic disease in which red blood cells do not bind oxygen properly. So they take on a sickle shape rather than a disc shape, and then they become trapped in the capillaries. So shown here is kind of, I'll move this a little bit. I got to make sure that my computer captures my voice, so that way that gets captured too. So, okay, so here is a normal red blood cell, and it's got this disc-like shape, and when everything's normal, they flow very nicely through capillaries. But then when they get this sickle shape, they actually start to kind of aggregate, and then they start blocking the capillaries. And so they block blood flow to the tissues, which then also blocks oxygen flow, or oxygen delivery. So this disease is actually caused by mutation of one amino acid, which is near the N terminus of the beta chain of hemoglobin. That mutation is where a valine of the polypeptide is actually mutated to, where glutamate is mutated to valine. And so this mutant is called hemoglobin S. Okay, and so when we had mutation of this residue, when we have this mutant, what happens is that glutamic acid is usually on the outside of the protein. Okay, and that's where it's interacting with its hydrophilic environment. Well, now when we mutate it to valine, the valine is now on the outside of the protein. And then what happens is that another hemoglobin will come by, which is also mutated, and those valines will actually stick to each other. Okay, so you actually have associations of hemoglobin molecules that are not supposed to happen. So when you have association of these valines through hydrophobic interactions, we then get this weird distorted shape of the hemoglobin molecule. This results in the sickle shape that we see in red blood cells, which then leads to the lack of oxygen in the extremities and pain, and is really the biochemical basis for sickle cell anemia. So one thing that's really interesting is that people that are heterozygous for the sickle cell gene, so they have one normal copy, but then they have one mutant copy, they actually have resistance to malaria, right? So I just said that having this gene is really bad unless you're one of these people that are heterozygous for the sickle cell. So it's been shown that in these regions of the world where there's a high incidence of malaria, there's more people that are heterozygous for the sickle cell trait. In fact, it can be up to 40% of the population can be heterozygous for the sickle cell trait, and that's because it allows them to have resistance to getting malaria, which I think is really interesting. So anyways, this is how, even at the primary sequence level, that is very, very important to protein structure and function. All right, so now we're going to talk about the secondary structure. So this is the three-dimensional structure of the local segment of the protein. And really, when I'm talking about secondary structure, I'm really talking about alpha helixes and beta sheets, the type of interactions that are occurring to make alpha helixes and beta sheets. And this arises from the fact that there's hydrogen bonding from the main chain atoms. So the carbonyl carbon is going to be in a hydrogen bond with the hydrogen of the nitrogen that's in the bond. So This is the carbonyl carbon, and this is the amide hydrogen. So the alpha helix. This is the most common secondary structure in proteins, and it's really common because it's super stable. And I mean, that's really what proteins are looking for, is how do I become the stablest protein possible so I can actually exist. And so it's rod-like. Well, and first of all, we call it the alpha helix because if you look at the ribbon diagram of the alpha helix, it's a helix. So it's rod-like. It's a very, very dense structure and involves only one polypeptide chain. So this is kind of the ball and stick model of the alpha helix, and you can see that's a pretty dense structure. And then it's stabilized by hydrogen bonds that are parallel to the axis. So there's hydrogen bonds that actually go, so if this is the axis of the alpha helix, the hydrogen bonds are going up and down the helix. And so what it looks like is that there's a carbonyl carbon, so the oxygen of the carbonyl carbon will hydrogen bond with the hydrogen on a nitrogen of the amide nitrogen 4 amino acids away. So if this is 1 amino acid, then 1, 2, 3, 4, it's going to hydrogen bond with the amide nitrogen, the hydrogen of the amide nitrogen 4 amino acids away. And this happens with every single amino acid in the polypeptide chain. So we have hydrogen bonds going up and down through this whole entire helix. And so, oh, go ahead. Yeah, yeah, exactly. It's the hydrogen bonding throughout the entire structure that actually makes it go into this helix. So the helical, and it's vice versa. It's the helical conformation that allows the linear arrangements of the atoms, giving the bonds maximum strength and conferring stability. And so per turn of the alpha helix, there's 3.6 amino acid residues. So in the alpha helix, all of the side chains, they're going to lie outside of the helix because there's not enough room in the interior. So this is the structure. This is actually the structure of hemoglobin. This is a ribbon diagram. And it's kind of hard to see, but do you see these little green sticks that are kind of sticking out on the sides? Those are the side chains. Those are the R groups that are sticking out of the helix. So if the alpha helix, okay, so proteins, they have varying amounts of the alpha helix, and it varies from just a few percent to none to 100%. And there are a number of factors that can disrupt the alpha helix. One of them is proline. So if there's a proline in a sequence, this is a helix breaker. So it actually creates a bend in the backbone because of its cyclic structure. And this is because the rotation around the bond between the nitrogen and the alpha carbon is restricted due to the ring kind of like bonding with itself back onto the main chain atoms, and the fact that in a polypeptide or a peptide, the proline's alpha amino group cannot hydrogen bond, cannot do interchain hydrogen bonding. The other thing that can destabilize an alpha helix is electrostatic repulsion. So if you have, say, side chains with the same charge right next to each other, this is unfavorable. So for example, two lysines, right? Those are both positively charged. If they're next to each other, that's not a very, favorable thing. So it'll destabilize it. Or if you have, say, two negatively charged amino acid groups, like two glutamates or a glutamate and a aspartate, it would destabilize it, as well as steric crowding. So if you have like these really big, bulky side chains next to each other, they're crowding each other. They're taking up each other's space. And so that's also unfavorable. So things like, say, bigger amino acids like valine, isoleucine right next to each other might not be favorable, or tryptophan and phenylalanine may also not be favorable, in the alpha helix. All right, so the beta sheet, so there's different names for, there's like different names for everything. So I apologize when I do use different names for everything, but there's lots of names for the beta sheet. So they called the beta cleavage sheet, also called beta strands. And this can involve one or more polypeptide chains. So I kind of mentioned how in alpha helix, it was just one polypeptide chain that created the alpha helix. Here, it could be one or more. And so the peptide backbone is almost completely extended, and it has this zigzag or pleated pattern. And so it's kind of like when you have, those paper fans, and you kind of open it up. That's what an alpha helix looks like. The hydrogen bonds then occur between different parts of a single chain that is double backed on itself, so there can be interchain bonds, or between different chains. So these are interchain bonds. So in the hydrogen bonds, they're going to be perpendicular to the plane. So here's the axis of the beta sheet, and our hydrogen bonds, they're running perpendicular. Okay, and so these can be parallel or anti-parallel. And so what I mean that this can involve one or more polypeptide chains. All right, so then it can be parallel or anti-parallel. So a parallel beta sheet means that the peptide chain runs in the same direction, and they're aligned in terms of the N and C terminal chain. So you have the two hydrogen bonding segments of beta sheets that are, they're going from N to C here and N to C here. And so and one thing to kind of note is that the hydrogen bonds between these, the carbonyl carbon and then the hydrogen of the nitrogen, they're kind of staggered. So they're not as strong as if they were like directly aligned. So these are not as common as anti-parallel beta sheets. So anti-parallel beta sheets are when the chains are running in the opposite direction. So we have N to C going in this direction, and then N to C going in the opposite direction. Okay. And so in this arrangement, the hydrogen bonds, they're like directly across from each other. And so this actually is much more stable than the parallel arrangement. So this tends to be a more common feature that we see. All right, so okay, so now we're going to talk about back to reverse turns or beta turns or hairpin turns. So these are going to be turns essentially that are going to connect the secondary elements. So protein requires that the peptide backbones and the secondary structure to be able to change directions. And so you need to have these different turns to transition between one secondary element to another. So oftentimes they will contain a proline, okay, which will help to create that bend in the turn, and then there's a glycine right next to it. So a very common type of turn is called a type 2 turn, and there's a proline, in the turn, so that's going to create the bend, and then the next amino acid is going to be the glycine. So it's just small enough to actually fit into the very little space that there is in the turn. And there's also, along with these different turns, there's also small areas of unstructured regions, which we call loops, or floppy regions, or linkers. So super secondary structure, these are just another permutation of a secondary structure, and this is where you have combination of secondary elements that come together, and we call them super secondary structures because they're very common in protein structures. So they're formed by a combination of alpha helixes and beta sheets, and they are not the entire protein, but they could be a part of it. So the most common one is the beta-alpha-beta unit, or also called, it's just a beta-alpha-beta unit. And so this is kind of the representation of it, where you could have two parallel beta sheets that are connected by an alpha helix. And another common structure is the alpha-alpha unit, also called the helix-turn helix, where you have two helices that are connected together by, a turn. And there's many other that are possible. Yes? Yeah, there is. Yep. Yeah, this is kind of, it's hard to kind of see it because this is a two-dimensional thing. But yeah, if you were looking at this, the beta sheets would actually be on the same plane, and then the alpha helix would be kind of like in front. Does that make sense? Oh man, I'm getting such great questions. I hope we can get through this whole talk. All right, so tertiary structure. There's two types of protein conformations. There's fibrous and globular types of structures. And so the nature of the side chains in a protein, which is part of the tertiary structure, can influence the folding of the backbone. And so one type of tertiary structure or type of tertiary proteins are those that are fibrous, so the fibrous proteins, they are compact and they're generally insoluble and they often play a structural role. So they can be alpha helices or contain beta sheets. And examples of fibrous proteins is collagen, so that's kind of the flexible, strong protein that's in skin and bones. Keratin, so that's in hair, which we know is very strong, as well as in wool. And then fibrin, which is in silk. So they can be, again, they can be alpha helical or beta sheets. So we're just going to talk a little bit more about the collagen triple helix as an example of a fibrous protein. The collagen triple helix is in bone and connective tissue, and they're water insoluble rod-like fibers of great strength. So they kind of form these rod structures. And so the collagen triple helix is 3 helical polypeptides that are wrapped around each other like a rope or a triple helix. And so it's kind of like you have these 3 protein strands that are braided together. And when you braid something together, it's a lot stronger. And each of the 3 chains has a repeating sequence of 3 amino acids, which is X, which can be like any amino acid, or it stands for any amino acid, proline followed by a glycine, or X followed by hydroxyproline followed by glycine. And then every third position is occupied by glycine, and this is where the chain is like crossing over, and so you need a little amino acid there in order for it to do that slight turn to cross over inside the helix. So what actually holds these 3 strands together is the hydrogen bonding of hydroxylated proline and hydroxylated lysine. So here's proline here, and it can actually become hydroxylated in the side group. And same thing with lysine, it can be hydroxylated at the gamma carbon of the side group of lysine. And so you can have hydrogen bonding interactions that can happen between these side chains in the triple collagen helix. And that's what really gives it that really, really strong structure. So one problem that can happen is that from not having very good collagen is a disease called scurvy. Who's heard it before? Some of you have, but so there's an actual biochemical basis of why this happens. So this disease results from a deficiency of vitamin C. Vitamin C is actually a cofactor required for hydroxylation of proline and lysine. And so if you have lower amounts of hydroxyproline, you're going to have less hydrogen bonding, and therefore the collagen structure is not very strong. And so the symptoms of scurvy, such as bleeding gums, sunken teeth, skin discoloration, is a result of having fragile or weak collagen due to not having very much hydrogen bonding. And so this is just a very famous example, is that a lot of the men on Columbus's ship that sailed from Europe to the Americas, a lot of them actually got scurvy because they didn't really have like fresh food, like fresh fruits to eat. They weren't getting their vitamin C, and so their collagen was very weakened, and they were getting scurvy from being on the voyage. So you can tell that to your mom and dad next time on Columbus Day. All right, so we're going to talk about globular proteins. This is a much more common type of tertiary structure, and this is when the backbone, just like it sounds, it's globular in shape. So the backbone folds back on itself to produce a much more spherical or globular shape. And so you can have, this could be, it can be different combinations of helical and pleated sheets that are arranged so as to bring everything really close together in 3 dimensions. So these are water soluble and they have compact structures, and they often have catalytic roles. So for example, enzymes are often soluble or globular proteins, as well as hemoglobin and myoglobin. So protein folding. So we have all of these different structures that's going to drive that's going to come together to give the protein its final fold. So the conformation of the backbone is going to put constraints on the possible side chain arrangements. The primary structure, this is the order of amino acids in the polypeptide chain, and it depends on the formation of covalent bonds. As we get to the higher order levels of structure, such as the conformation of the backbone in the secondary structure, and then the positions of the atoms in the protein in the tertiary structure, this depends on the non-covalent interactions that's going to drive the formation of a secondary and tertiary structure. And then if the protein consists of several units, such as in a quaternary structure, this also depends on non-covalent interactions. So the take-home point is that non-covalent stabilizing forces is what contributes to the most stable and lowest energy structure. And so folding can bring together residues that might be far apart in sequence, closer together, and this can be influenced by environmental factors. All right, so forces that stabilize the tertiary structure of proteins. The major driver of stabilizing tertiary structure is hydrophobic interactions. Okay, this is like the primary driver of it. If anyone tells you it's not, tell them that they're wrong. It's hydrophobic interactions. So this is when nonpolar residues cluster together in the interior to escape contact with water. So this is driven by thermodynamics. What does that sound like? Yeah, right, exactly. This is the same thing. We're putting all the nonpolar stuff in the interior, and then we're putting the polar stuff on the exterior, right, to avoid contact with water. So the same forces that are driving micelle formation is driving protein folding in the tertiary structure. You can also have hydrogen bonding between the side chains of amino acid as well as between side chains of the backbone. That's also an important driver. Still not as important as the hydrophobic interactions. And then also electrostatic interactions between oppositely charged groups, or as well as in contrast to repulsion of the like charged groups. And then sometimes you can also have binding of the protein to metal ion cofactors or to prosthetic groups, and this also helps to get it to fold into the right conformation. So in addition to non-covalent interactions, disulfide bonds form covalent links between the side chains of cysteines, and this can severely restrict the available conformations. Okay, yeah, so, and then the tertiary structure is often determined experimentally, so using X-ray crystallography and 2D NMR. And if you're interested in knowing what that is, I can tell you some other time. So not all proteins exhibit all interactions, and then with really any protein in water, nonpolar residues are going to be on the interior, and then the polar residues are going to be on the exterior, just like we discussed about micelles. Okay, so myoglobin. We're going to talk about myoglobin as an example of a tertiary structure because it's actually really quite interesting, and there's a lot that's known about it. So myoglobin's function is to bind oxygen in the tissues. It stores oxygen in the tissues. So in a structure, it contains a heme prosthetic group. So this is what the heme prosthetic group looks like. And so the heme is kind of bound into this hydrophobic pocket of myoglobin. It's got 8 alpha helical regions, so it's mostly alpha helical. It's 75% of the protein. And then, as should be for globular protein, the side chains are on the exterior, and then the nonpolar residues are in the interior, except 2 histidines. There's actually 2 histidines that lie in the interior of the protein. So when you have like something kind of funny, like a charged amino acid in the interior of a protein, which it seems like it shouldn't be there, that's an indicator that it's probably doing something important for the protein's function. So again, the heme is planar. It fits in the hydrophobic pocket, and the heme group is iron that is bound to these protoporphyrin rings, so these 5-membered rings. So they're all linked together, and then they bind iron. So iron has 6 coordination sites, 4 of which are occupied by the nitrogens from these porphyrin rings. The 5th coordination site is coming from that histidine. So one of the histidines are actually coordinating or bound to the iron. And then the 6th is oxygen. So the 6th coordination site is where oxygen binds to iron. So the 2nd histidine in the structure, it acts as kind of a gatekeeper. So it's going to open and close to allow oxygen to enter the pocket. And so this is what it looks like when oxygen is bound to the heme. The histidine is kind of there, and it causes the oxygen to kind of bind at an angle. So it's an imperfect binding that happens. So, why would there be this imperfect binding? Because it's not as strong as it would be if the oxygen was perfectly binding linearly to the heme. The reason why, or at least it's been postulated, is because the heme group can actually bind other molecules, like carbon monoxide. So, what happens what do you guys know about carbon monoxide? What was that? Carbon monoxide. It can kill you. And the reason why it does that is that it will actually, if it binds, it will outcompete the oxygen. So then now, and then your myoglobin isn't able to deliver, it outcompletes oxygen so that way your tissues aren't getting oxygen because now we have carbon monoxide binding. Okay, so now we're going to talk about why this imperfect binding is beneficial. So if myoglobin wasn't there and we just had that heme group, or if the histidine wasn't there, then the carbon monoxide could bind perfectly to the heme group. And so this would mean that carbon monoxide would bind 25,000 times more tighter to oxygen if the histidine wasn't present. And so by having that histidine there and causing imperfect binding of the carbon monoxide, it's only 250 times stronger if it's binding at an angle. So it's not quite as bad as it could be. So it's really bad. I certainly wouldn't want to turn on my car in my garage and shut the door and stay there for a long time. But at least this kind of helps a little bit of making sure it's the heme group is binding to oxygen and not carbon monoxide. All right, so denaturation and refolding of proteins. Weak non-covalent interactions can be disrupted, destroying the three-dimensional arrangements of atoms. When we talk about denaturing a protein, we mean unfolding it. So we mean unfolding it. And so when we denature it, the primary structure, that is the covalent amide bonds, they stay intact, and the non-covalent interactions are disrupted. So this is just an example of a native structure, the native folded protein structure. When we denature it, it has some unknown denatured structure. We don't really know what it looks like. All we know that it does not look like the folded structure. And it also is not functional once it's denatured. So what's really interesting is that under experimental conditions, the disrupted structure, we can take this denatured structure and we can actually refold it back again to have a native conformation and to have all of the activities that it's supposed to have for some proteins, not all of them. And this is evidence that primary sequence contains all the information required to produce the three-dimensional structure. So, denaturation and refolding of proteins. Proteins can be denatured in several ways. One is heat, or sonication. So if you apply heat to a protein, say 5 to 10 minutes at 95 degrees Celsius around boiling point of water, this increases the energy and vibrations of the bonds and disrupts the tertiary structure. pH extremes, they can also cause proteins to denature. And so, one of the things, so this is going to affect the ionization of functional groups and alter electrostatic interactions. So this is kind of related to all of these ionization of amino acid titration curves that we're working on, in the previous lectures. And salts can also interfere with ionic interactions. Detergents, such as sodium dodecyl sulfate, so SDS, they disrupt hydrophobic interactions. So here is the structure of SDS, so sodium dodecyl sulfate. Does that look like anything that you guys have seen before? What's that? It does. And what's similar about them? Yeah, but is there something in terms of like polar or nonpolar? Yeah, it's amphipathic, right? So it's an amphipathic molecule. So this is okay. Urea alcohols and guanidinium HCl or hydrochloride, they form with hydrogen bonds with a protein that are stronger than those with a protein itself. So they disrupt intramolecular hydrogen bonds. So basically, they outcompete the existing hydrogen bonds in the protein because the molecule themselves can form hydrogen bonds with a protein. So you can see this is the structure of urea, which has lots of areas where you can have hydrogen bond take place and disrupt the existing hydrogen bond in the protein. And then beta-mercaptoethanol, this is a structure. It's got a thiol group on it, so it's got a reduced sulfur on it. It will reduce disulfide bridges to 2 sulfhydryl groups. So we're going to talk about prions for a couple minutes, and then I'll give you guys your break. But one really interesting thing that can happen as a result of protein misfolding is prions. So how many of you guys have ever heard of prions? Yeah, right. Like it's been kind of in the news recently. So prions are the causative agent of mad cow disease, as well as scrapie in sheep and spongiform encephalopathy. So in humans, and it is a small 28 kilodalton protein, which is a glycoprotein that is found in cell membranes of the tissue. So the disease arises when the normal form of the protein, called PRP, this is the structure of it, it's more alpha helical, it forms into the incorrect form called the PRPSC. So it actually converts to this structure, which has more beta sheet than alpha helix. So it's the same protein, it's just it can fold into a different structure. And so when we have this abnormal prion with a beta sheet, it can actually make more of itself. It can actually propagate normal forms into the abnormal forms. And if it does that enough, and so it'll keep propagating and eventually reach the nervous tissue and the brain. And so once you have enough of it, then enough of these beta sheets, they can actually start aggregating and form these insoluble plaques. And so this is also a common fate that we see in Alzheimer's disease. It doesn't quite use prions, but it's a different type of protein, but it's the same thing where you have proteins that are folding, and I guess they're misfolding in the incorrect way to then aggregate and cause these insoluble plaques. And so this can cross species barriers. So if you eat tainted beef from animals with mad cow disease, you can get the spongiform disease. Okay, so quaternary structures. So first, let's just talk about nomenclature. So the name is going to be based on the number of subunits or the number of polypeptides it has. So if it's in the whole structure, if it's got 2 polypeptides, it's a dimer. If it's 3, it's a trimer. If it's 4, it's a tetramer, and so on. And then we also talk about whether it's the same type of polypeptide or different types of polypeptide. So homo means that you have like the same type of polypeptide. So if it says like a dimer and it's a homodimer, it would mean that the 2 polypeptide chains are exactly the same in amino acid sequence. And then hetero mean that they're different. So they're 2 or more different polypeptide chains in the quaternary structure. And then in terms of naming the specific subunits, if there's it goes from alpha, beta, gamma, and delta, and so on. So just to kind of give you guys an example, hemoglobin is a heterotetramer, so it's got 4 different subunits, and it has 2 each of 2 different subunits. So it's got 2 alpha subunits. Here's one. Here's the second one. So their amino acid sequence are identical. And then it's got 2 beta subunits. Here's one, and then here's the second one. So those amino acid sequences are identical. So it's a heterotetramer, and we would say that this is alpha 2, beta 2. Here is another protein. This is called BPS75. This is it's a protein chaperone, so it likes to go and find other proteins and chaperone them around, kind of like a parent chaperone. And so it's a homodimer, so it's got 2 alpha subunits, so they're exactly identical to each other in terms of their primary sequence. So we say that they're alpha 2. Yes? So now we're going to talk about hemoglobin as an example of a quaternary structure. This I think is so cool. So hemoglobin, it's got 2 alpha subunits and 2 beta subunits. So the alpha has 143 residues, the beta has 146 residues, and these subunits are conserved. They're very similar to each other in terms of the amino acid sequences. So the alpha subunits are actually very similar to the beta subunits, even though they are actually different, but they're very similar. So there's regions of conservation and homology as well as identity and similarity. And just to mention that it also has conservation with myoglobin. So they're very similar in the sequence as each other as well as the sequence of myoglobin. For hemoglobin, each subunit of hemoglobin binds a heme group. So here's one here, here's another here, and here's the third and fourth. And so each heme group is a binding site for oxygen. So there's 4 subunits, 4 heme groups, and then the ability to bind 4 oxygen molecules. And so you can have different quaternary structures depending on whether hemoglobin is oxygenated or deoxygenated. So if hemoglobin is oxygenated, so oxygenated hemoglobin, it's going to be bound to O2, and that conformation has a high affinity for oxygen. So it wants to bind more oxygen. For deoxygenated hemoglobin, it has a different conformation, so oxygen is not bound, and it has a low affinity for oxygen. So it does not want to bind oxygen. And so it's actually structurally different. So oxygenated hemoglobin kind of has a much more relaxed structure, whereas the deoxygenated hemoglobin has a much more tight or tense structure. So one thing about hemoglobin is that it can be allosterically regulated, which means that changes in the shape of one site causes conformational change at a distant site. So if oxygen binds to hemoglobin, it causes the whole conformation to change. So we have one molecule of oxygen binding at one site, and then this at one subunit, and then this will get transmitted through the entire rest of the molecule. So, so this is what we call positive allosteric regulation, in that oxygen binding actually increases the affinity of hemoglobin binding oxygen. It's kind of like a chicken and egg problem. Okay, so we're going to talk a little bit more about that because I know it's a little bit confusing. So let's talk about comparing myoglobin and hemoglobin. They're very similar in many ways. They both bind oxygen, they both bind heme groups, so they have very similar primary, secondary, and tertiary structures. But the difference is the quaternary structure. So hemoglobin actually has a quaternary structure, so it's got the 4 different tertiary structures that assemble together, whereas myoglobin only has just the 1 tertiary structure. So there is no quaternary structure for myoglobin. So myoglobin has got 1 subunit, 1 heme group, binds 1 oxygen. That's the structure of it. The function is to store oxygen in the tissues. Hemoglobin has 4 subunits, 4 heme groups, 4 oxygens, where it can bind 4 oxygens. And so it has a different function, which is to pick up or bind oxygen in the lungs and then deliver it to myoglobin in the tissues. So it's going to deliver oxygen to the tissues. And the question is, how does structure dictate function, and how is it reflected in its oxygen binding properties? Sorry, the answer is due to the fact it's either 1 subunit or 4 subunits. So we're going to first talk about myoglobin. So myoglobin's function is to store oxygen in the tissue. And so this is the oxygen binding curve of myoglobin. On the x-axis, we have the oxygen pressure, so the partial pressure of oxygen in pores, which is essentially the oxygen concentration. And then on the y-axis is the degree of saturation. So what that means is what fraction of myoglobin is bound to, what fraction of myoglobin is bound to oxygen. So this can, range from 0 or 100% to completely bound, or 0% to 100%. So a fraction of 0 to 1. And so this is oxygen binding curve of myoglobin, is that as the level of oxygen increases, the degree of saturation, that is oxygen binding, sharply and steadily increases until there's complete saturation. So myoglobin is completely bound to oxygen. So this generates a hyperbolic shape of the oxygen binding curve. And this is due to the fact that there is just 1 subunit of myoglobin that can bind to oxygen. And so as a result, myoglobin has a high affinity to oxygen in the tissues. So if we kind of compare the concentration of oxygen in the lungs versus the tissues, we can see in the tissues it's about 10 torrs. So we kind of go up, we can see that approximately 100% of myoglobin is bound to oxygen. So now it's serving its role, which is to bind to oxygen and carry out its function of oxygen storage. So now this is really quite different to what's happening in hemoglobin. With hemoglobin, it's a very, very different story. So the function of hemoglobin is to bind oxygen in the lungs and then deliver that oxygen to myoglobin in the tissues. So what does the oxygen curve look like? With hemoglobin, it displays cooperative properties, which means that each of the 4 oxygen molecules bind with successive ease due to conformational changes. And so the binding affinity of the first one is lower than the binding affinity of the second one, then the third, then the fourth. So what that means is that if hemoglobin is not bound to oxygen, so it's the deoxyhemoglobin, it is really, really tough for that first oxygen molecule to bind. But then once that one binds, then it's kind of gone through a conformational change where now it's easier for the second one to bind. And now that the second one's bound, it's even easier for the third and fourth to bind. So that's what we call cooperative property or cooperative binding of oxygen to hemoglobin. And so this generates a sigmoidal shape to the oxygen binding curve. So you can see that, at low concentration of oxygen, it's really, really tough for that first one to bind. And then, so this curve is relatively flat, so it's got a very shallow incline to the curve. And then as the levels start to increase, and as more oxygen starts to bind, the slope of this curve starts to increase until we achieve 100% saturation at high oxygen levels. And this is due to the fact, again, that there's the four-sluggedness of hemoglobin. So, as a result, in the lungs, there's high level of oxygen, so there's a high level of saturation of oxygen to hemoglobin. And then in the tissues, there is low levels of oxygen, so there's low levels of oxygen saturation to hemoglobin. And so this is what really allows it to carry out its function. So it allows hemoglobin to pick up oxygen in the lungs and then go to the tissues where now it's got the low affinity where the oxygen is lower, and then deliver the oxygen to myoglobin in the tissues. So basically, hemoglobin binds to oxygen when the concentration is high, and then it releases it when it's low. All right. So there are different things or factors that can affect the binding properties of hemoglobin to oxygen. So one of those is pH changes. So the pH of the blood and surrounding tissue can influence the oxygen binding properties of hemoglobin. And so what happens is that as the proton level concentration increases, so the pH gets lowered, this actually reduces the affinity of oxygen to hemoglobin. So this gives a binding curve with an even more sigmoidal character. And so as the pH decreases, the percent oxygen saturation also decreases. And so this just shows the binding curve of hemoglobin at different pHs. And you can see that as we decrease the pH from 7.6 to 6.8, the curve actually becomes even more sigmoidal. So there's less oxygen bound to hemoglobin. So, what does that mean? Like, why do I care? Well, there's an actual physiological role for why the pH can affect hemoglobin binding. And so this is called the Bohr effect, in which the effect of the proton concentration on where there is an effect of the proton concentration on the oxygen binding. So in the tissues, the pH is lower, and recall that tissues, through metabolism, they produce carbon dioxide. So they produce CO2. And recall that when carbon dioxide is in water, this makes carbonic acid, which carbonic acid can dissociate into bicarbonate and then hydrogen ion. And so this actually increases the hydrogen ion in the solution, which will then lower the pH. So when this happens, having this lower pH, the hydrogen ions will then protonate, key amino acids like histidine 146, which are then stabilized to a negatively charged salt bridge, aspartic acid 94, which then favors the deoxygenated or the low affinity state of hemoglobin, and then causing it to release oxygen. So I'm just going to illustrate what that looks like. So that's what happens. Okay, so in the tissue, the pH is lower. So then, we have this stabilizes the low affinity form of hemoglobin, and then it's going to release its oxygen to the tissues. But so then when we get to the lungs, the pH is higher, and so this reverses the effect. So this is no longer going to be in a salt bridge. The histidine will not be protonated, and so the salt bridge cannot form, and so that will destabilize the formation of this low oxygen affinity form of hemoglobin. So then, together this allows hemoglobin to perform the function of picking up oxygen in the lungs at high pH and then unloading it to the tissues at low pH. Here's just a summary for what happens in the Bohr effect, basically everything I just said. And then the last thing I'm going to talk about is another regulator of hemoglobin, so another allosteric regulator, which is called 2,3-bisphosphoglycerate, so it's BPG. And this, like I said, this is a negative allosteric regulator, which means that it's going to prevent or inhibit oxygen from binding to hemoglobin. So this is 2,3-BPG right here, so bisphosphoglycerate, and it's got all these negative charges. So it's coming from the carboxylate group and 2 phosphate groups. And what it does is it stabilizes the deoxygenated form of hemoglobin. So it stabilizes the low oxygen affinity state form of it. And the way that it does that is that in the cavity of the deoxyform is this cavity. And this cavity has basic residues. So they are positively charged. So there's like histidines and lysines and all these different charged residues that are positively charged in this cavity. So what happens is 2,3-BPG will bind right inside this cavity. So it's going to form these salt bridges or these ionic interactions between the side chains of the deoxyhemoglobin and then bisphosphoglycerate. So that binding stabilizes the low affinity state or the low affinity conformation of hemoglobin, which then, also facilitates the release and delivery of oxygen to the tissues. So this is something that, it's happening in all of us right now. We have 2,3-BPG in our blood, and it's part of what's causing the sigmoidal behavior of hemoglobin. So there is a role in 2,3-BPG in delivering oxygen to the fetus from a pregnant mother. So there's an actual biochemical basis for this. So the fetal hemoglobin is actually different from adult hemoglobin. So instead of having the beta-2 polypeptide chains, it has a different polypeptide chain, which is called alpha-2. And so the difference between the beta and the gamma, sorry, I said alpha, I meant gamma. So the difference between a beta and a gamma is that it actually has a his to serine substitution. So instead of having histidine in this cavity, it's got a serine instead. And so what happens is that there's fewer ionic interactions with BPG, so it's not able to bind as well into the cavity of deoxygenated hemoglobin. So what happens is that this lowers the affinity of BPG to the deoxy form, which means that having less BPG bound will favor the high affinity state of fetal hemoglobin to oxygen, or it'll favor binding of oxygen to fetal hemoglobin. Did that make sense to you guys? Okay. So what does this look like? So again, as I mentioned, the fetal hemoglobin, it wants to bind, it binds oxygen tighter compared to maternal hemoglobin, and so this difference in oxygen binding is what allow oxygen to flow from maternal hemoglobin to fetal hemoglobin. So the fetal hemoglobin is going to extract the oxygen from the maternal hemoglobin because of this difference in oxygen affinity and the way that it interacts with 2,3-bisphosphoglycerate.